Okay, and our third speaker is Jada Nelson. And Jada's going to tell us about the effects of MCP1 chemokine on ovarian cancer cell viability.
1 second. Okay. So my title is a little different than what it was previously said. So it's MCP1 or CCL2 promotes migration and invasion of ovarian cancer cells by inhibition of AMPK. So I basically just wanted to start off simple. What is ovarian cancer? So ovarian cancer is cancer that arises in the ovaries, as pictured here on the right, and that's in the abdominal cavity of the female. And ovaries aid in the female reproductive system, and the way that they do this is they do egg formation, the release of eggs, which is ovulation, and also production of hormones. And so when all these processes are happening, sometimes you can have uncontrollable changes or abnormal growth that's occurring that can cause tumors to arise in the ovaries, as pictured here on the right. So why is ovarian cancer so prevalent right now? What's the 5th leading cause of death in females with reproductive diseases? And so the problem is most patients are diagnosed at advanced stages. And so usually they're diagnosed at advanced stages because there's not a lot of early detection, because the signs and symptoms are usually non-existent or very vague. And then also there's a difficulty in looking at the location. The ovaries are deeply embedded within the abdomen. And then also the size of the ovaries. To give you a comparison, they could be like the size of an almond. And then because of all these things, we don't have a lot of information about the unique biology of the disease. So because we don't have a lot of information about this unique biology of the disease, there's not a lot of treatments out there for ovarian cancer patients. So one part or aspect that my lab looks at is repurposing metabolic drugs as treatment. So one of them is metformin, and metformin is usually used in type 2 diabetic patients, and they saw that type 2 diabetic patients that were taking metformin had a lower risk of encountering cancer and also increased risk in survival. So the way that metformin works in the body is that it activates a kinase in our body called AMPK. And essentially what AMPK does is it regulates the metabolism and cell, or the energy and metabolism of every cell in your body. So if you have a dysfunction in AMPK, then you don't have checkpoints that are being checked for your cells to grow and regulate, or the energy and the nutrients. So what my lab wanted to look at is AMPK activity in correlation to tumor outcome. So they did an experiment where they used ovarian cancer mice and they put them on a high-fat diet and a low-fat diet. And so the picture on the left is data basically from the high-fat diet mice that they had a burden of aggressive ovarian cancer tumors. So if you look at the left image, all the purple areas signify tumor growth around different areas in the body and different tissues that they did. So like I said, they wanted to look at tumor outcome in relation to AMPK activity. So the picture on the right shows you they did a staining of the tissues. And so the brown coloring basically signifies AMPK activity, because that's the protein that we were staining for. And so as you can see in the regular diet tumors, there's a lot of brown areas, and that's basically showing you that there's activity of AMPK, versus in the high-fat diet tumors, there's not a lot of brown areas, so we're seeing a reduction in the AMPK activity. So then my lab wanted to see, well, what proteins or what factors affect the high-fat diet tumors that reduce the activity of AMPK. So they did an in vitro study, and they took ovarian cancer cells and they exposed them to fat conditioned media. And what they saw was they had an upregulation of certain proteins. So the picture on the left shows all the proteins, and one of the highest ones they had was CCL2, and that's the protein I'm going to be talking about. It's also called MCP1, and it stands for monocyte chemoattractant protein. And MCP1 essentially is produced by tumor and immune cells, and it aids in white blood cell migration and also inflammatory response. So basically what happens is when you have the tumors are present, they have receptors, and then MCP1 binds to those receptors, and its preferred receptor is CCR2. It can migrate throughout the body, adhere to different tissues, and then cause invasion. So then our hypothesis is that MCP1 promotes migration and invasion of ovarian cancer cells by inhibiting this AMPK protein. And basically this is just a chart to say normally when AMPK is functioning normally, you have those specific cell checkpoints. And it's inhibiting ovarian cancer cell migration. And what we believe, based on that MCP1 aids in migration factors, that there's a direct correlation between MCP1 and ovarian cancer cell migration. And so then we want to look at, well, how does MCP1 do this? So it binds to its receptor, and then we're believing that it's inhibiting this AMPK. And then we also want to look at, well, if it's inhibiting this AMPK by binding to its receptor, how is it doing that? So the first experiment we did was we wanted to see if MCP1 was causing growth of ovarian cancer cells. So we did a MTT assay, which is essentially looking at if the cells are growing or if they're not growing. So we had two different conditions. We exposed the cells in serum and serum free. And so serum is just essentially nutrients that you can add. So with nutrients and without nutrients. And so basically when we did this, our data wasn't very consistent across the board. So as you can see in the 24 hours, we kind of see a decrease and then an increase. But at the 48 hours, it's not very consistent. And with serum, so with nutrients, and then without nutrients, we kind of see a trend at the beginning in the 24 hours. We could maybe infer that as MCP1 concentration increases, that the cells have a higher percentage of viability, but it wasn't consistent at the 48 hours. So basically what we said was the protein of MCP1 doesn't necessarily have a significant role in proliferation. But based off of that, then we continued to look at migration factors. So we did another experiment, and it's called a scratch assay. And a scratch assay is basically looking at the migration factors of MCP1. So what we did was we took a 6-well plate, and we plated ovarian cancer cells and made them fully confluent. And essentially confluency is just a high concentration of ovarian cancer cells. So we took a pipette tip and we made a scratch in each plate, and the scratch created a gap, as you can see. So in these pictures at the bottom, the open spaces would be the gap. And so then we treated the ovarian cancer cells with MCP1 because we wanted to see if that MCP1 protein was causing migration factors of ovarian cancer cells. So then when we plated with MCP1, we did a low dose and a high dose, and then we took pictures of the course over the 3 days to see how the gap was closing. And the gap closing signifies that the cells are migrating. So as you can see in the bottom, there's a control 50 nanograms and 100 nanograms. And so our results state that although the control was migrating over the course of the 3 days, with the addition of the MCP1 protein, we can see that the cells are migrating at a faster rate regardless if there's a low dose or a high dose. So then at this point, we've confirmed in our hypothesis that MCP1 causes migration and invasion of ovarian cancer cells. So the next thing we want to look at is how is it doing this? And our hypothesis was that it's inhibiting AMPK. And so to prove that, we did a Western blot, and essentially what you're doing is identifying specific proteins. So if you look at this image on the right-hand side, you can see that there's a line that says PAMPK, and that's essentially what we're looking at. We're looking at the AMPK activity. And then the experiment was set up as control, MCP1, control, and MCP1. And control is just basically samples that have no treatment at all. And then MCP1 are samples that were treated with this particular protein. So when the samples were treated with this particular protein, we want to look at the band intensity. And so, as you can see, when cells were treated with MCP1 treatment, regardless of the time interval, that the band is smaller. And so the band being smaller indicates that there's a reduction in activity of AMPK. And once again, AMPK is that cell regulation checkpoint of ovarian cancer cells. So then at this point in our hypothesis, we confirm that MCP1 is aiding in migration and invasion of ovarian cancer cells by inhibition of AMPK. So previously I talked about how our lab saw that metformin activates, the way that metformin works is it activates AMPK. So we kind of wanted to do a controlled kind of assessment with metformin to say that we know MCP1 inhibits or causes migration with inhibition of AMPK. If we add in metformin, then we should see the reverse effect. We should see that there's not as much migration with the presence of metformin. So basically our results show that we did another migration assay. We took the plates, we made the gap, then over the course of the 3 days we took pictures, and then we did the low dose and the high dose as well. And so then we saw that this graph shows that with MCP1 treatment there's an increase in migration, but with the addition of metformin there's a decrease in migration. So then this slide basically just summarizes all the things that we did, so that MCP1 promotes migration of ovarian cancer cells at a faster rate regardless if MCP1 is treated at a low dose or a high dose compared to the control, but MCP1 does not affect the proliferation or growth of ovarian cancer cells. And then we also showed that MCP1, the way that it aids in migration and evasion is it reduces the activity of AMPK. And then the last one, we did the activation of AMPK with metformin, and it shows that metformin inhibits MCP1 by reactivating that AMPK or that cell regulation. So then my last slide is just basically talking about what's next, what are we doing next. So we did the migration assay to look at how the cells are migrating, and just to confirm that, we are going to do a transwell assay. And then also, I previously talked about, we know that when MCP1 is bind with this receptor, it's inhibiting this AMPK, but we don't necessarily know how it's doing that. So we want to look at this particular pathway that's highlighted in the yellow, and the way that we're going to do that is we're going to use the 88 gene array panel that covers the genes that are involved in migration and invasion. And so then we're going to look at, well, what genes are regulated by MCP1, and when those genes are regulated by MC1, how does it inhibit or modulate AMPK activity? And then the last thing, a lot of the experiments that I did were in vitro, so we want to look, my lab is looking at the in vivo effect of this particular protein in mice. So then my last slide is just acknowledging, this is my research lab, the Rattan Research Lab. And then I also worked in a neurology lab over the summer as well. And so a lot of these people in this lab just helped me with the project. Also the Henry Ford Health System, that's where my lab is located, and University of Detroit Mercy and the ReBUILDetroit program.